in an area with lots of different sponges, but now also some corals. So it's quite nice to see all this structure being formed by animals in addition to the rocks. So, and we've noticed that we see animals either inside or in close proximity to the sponges and climbing up on some of these corals. So definitely other animals use sponges and corals as habitat in this area.
Yeah, so these corals and sponges that are basically biological structure, they form structure that other animals can use for shelter, like the fishes that you see here. And what many of these animals do, the sponges and the corals, they filter the ocean water and they capture plankton, microscopic organisms or very small organisms, and they feed upon them. So they need a place where they have some current and where they will not be covered up by sediment, because the sediment will clog their ability to use their filter feeding polyps. And this is a good area. The hard substrate is a good place for them to live.
Definitely, we see that there are also some fish here. Looks like we've got some, I'm hoping, thorny heads. I'm right on that one as well. At this depth, it's a little different. When we were diving deeper, I always knew they were thorny heads, but now I know we've got some different rockfishes mixed in here, and they can be a little difficult to tell apart.
We need Tom to help us relearn everything we learned about rockfish.
I know.
Deep.
Oh, yeah.
Oh.
So, Ben?
I know. I heard Lisa and Tom this morning. Okay, fill me in.
It's a real feel-good ID work we had.
Yes, yes. So it was the sharp nose skate, but we need to find one so that we can show off our ability.
The long nose skate.
But Jen, oh yeah,
long nose skate.
Short.
But Jen is very good at identifying a flatfish. So anything that we find,
I will keep my eyes out for flatfish. The coolest fish there are.
Yes. Yeah, I was all ramped up.
I think we're going over one. Oh no. Sorry.
Oh no, I was just gonna say it was you know, for those of you that are just joining us, wondering why it says we're exploring Central California National Marine Sanctuaries on Ocean Explorations Trust's page or Nautilus Live, we are actually south of Anacapa, about south-southwest 13 miles or so. And yeah, we had some weather issues earlier in the week. It's been really hard to dive up in Central California. So you're right, I was all prepared. We had our one big dive up there, familiar with a lot of things that are 3,000 meters deep. And now suddenly we find ourselves here and I'm like, sponge, fish, coral.
Depth matters.
Right, for comparison, we're at 374 meters instead of 3,074 meters, and those are very different parts of the ocean.
Yes,
very familiar with
kelp forests too, the really shallow and the really deep. Kind of in between on the slope, not as much.
Well, luckily Kurt and Diana are helping us out, saying that here will likely be split nose and blackgill rockfish with white spots. Oh, rockfish with white spots or subspecies Sebastomus. So that is helpful. And Diana Water says we've also been seeing a lot of bank rockfish. So that's all very helpful. Thank you. This is really interesting terrain here. This eroded rocky formations, and if I was a fish, I think I'd be interested in all the nooks and crannies where you can hide here. So it's understandable. I think we just saw a hagfish that had curled up on the seafloor. We just went over that. earlier today, there was some hagfish or was that yesterday? Hagfish and sponges. Have you seen any of those today?
We saw some this morning.
I've not seen the. Oh, okay. We in sponges, but I've seen them around the
Yeah.
sponges.
Oh,
But
not in, but
yeah, yeah. We've seen
Yesterday, there was
hagfishes,
definitely a hagfish inside a sponge.
And so, remind me, Ben, what did you think of them? You thought that they were what?
adorable, but
Yes.
I didn't have any encounters with the rasping teeth, so
Right. So
just from
he
a
heard from
distance.
Tom. Yes. From a distance, they're adorable. Yeah. Oh, that's
They are
amazing
remarkable.
sponge.
I'll say that. That is. Well, a couple different beautiful
Yes.
forms of sponges there. One with the more brownish, dirty coating on the outside, that's tubular, and then one that looks like almost like a quesadilla folded over there in the middle. And
That's a good one.
I love the quesadilla sponge. No.
Yes.
And then the one behind there with
Great
some
mix.
kind of ruffles along the edges or the sides. But if you look, we see lots of yellow sponges too. Oh, we just got a question in the science chat. Can you guess the sponge people nicknamed the IKEA lamp?
The one that we just went by.
Which one?
The one with the ruffles.
You think that's the IKEA lamp?
Yes, it looks like an IKEA.
When
lamp.
I don't have something that comes up in my mind as like the typical IKEA lamp, so I'm drawing a
And
blank.
Abby said yes. Because it does look like, I was going to say, when I see these sponges, and I've seen a couple sponges that have had this really beautifully situated crab sort of holding on to it, I thought that would really make an amazing lamp or vase. If somebody could create this, mass produce it, I think it would sell.
There you go. There is a lot of, design that's informed by nature.
Yes.
I know I enjoy when things have a natural kind of look to them, informed by nature. So there you go. Maybe someone's going to hear you and make that happen. That was a really nice sponge we just passed over, the big white one.
Yes.
And I don't know if people heard, but there's been so many different sponges that have been encountered that are on a list of that scientists are trying to better understand the many different species of sponges that occur in this area, and they're very hard to identify from video. So when the opportunity is presented where there's a sponge that might be one of those and the ROV can safely land and take a piece of it, we've been collecting parts of sponges that are going to go back to the lab and be looked at very critically. There's components inside them called spicules that can be used to help identify them as other aspects of their cellular structure and their composition, and maybe also their genetics that help to differentiate the species. And then once you can figure that out from having a sample in hand, it may be easier to then apply that knowledge to these video surveys, so that you can, over time, do a better and better job of differentiating and enumerating species along these video transects. Well, look at that rock, just covered in corals and many different kinds of sponges going by on the left side of the screen. Beautiful. Oh, there's a skate, Lisa.
Oh yeah.
Ask and you shall receive. Is that your requested
Yes.
species? All right. So now you can teach
Yes.
us what you learned.
And and what did you learn, Ben? Was that a male or a female, you think?
I didn't even see the claspers.
So if you didn't,
It's female, but I mean, I didn't pay good enough attention.
I didn't see them either, so I think it was the female. It's quite obvious. There's just sort of an extra lobe under so you have the large wings, and then there's sort of a lobe underneath there, and then you'll have, for the males, there'll be another lobe.
They're not hidden on the ventral side like in the other sharks.
But they're not used as wings.
We're consistently seeing this coral, like as we can see in the middle left of the screen. It's like a light pink color and branching, but almost two-dimensional. And I'm curious to know if if you, Lisa, or anyone else can tell us what species of coral that is, and if that's something that was also part of the collections that were made earlier today.
I do not know. It's not Paragorgia.
Bridge.
We'll see. Uh-oh. Someone
Yeah.
in science chat, Daniel, has said that we've missed 2 flatfishes already. We've been looking at the sponges and we've missed. I'll have to keep a sharper eye.
You have the video for later.
So we're also seeing on an earlier part of the dive, we were seeing a lot of dead sponges. I'm not seeing as many here as before, but when sponges die, because of the spicules that they have inside of them, they also, that framework remains and they become a pretty good sort of substrate or surface for other animals to settle upon and grow. So the death of a sponge can become the habitat for another sponge or coral. Okay, so the white one, Kurt let us know that that white branching coral that we just saw, that was a Paragorgian. Cool. And they are also known as the bubblegum coral. I guess the pink gives it away. Good name, bubblegum coral.
Yeah, I don't know, Lisa, did you see the video from some of the dives on Olympic Coast? They had some very beautiful, floofy pink corals up there. I loved that term, floofy. But all the polyps were out, and they just were really magnificent.
Here we have a very reddish pink coral on the right with a rockfish facing it, admiring it. That probably is a Paragorgid. Yep. And then the rockfishes with the sort of two white bands on their behind their eyes. Which rockfish is that? I'm asking Kurt. You can, or maybe Di. Okay. So here we see a couple of rockfishes. think there's two different species because they're not the same. One of them has more white dots sort of towards the end.
It seems like one has a wider head.
Yeah,
Like the one just
sort
down
of
to the left. Yeah.
lots of sponges still. And I think all of those branching white corals, those are all paragorgids. And there's some sea urchins there that we just
the pink
passed.
The pink pin cushions. So, on the seafloor. I think there might be some of those sea cucumbers as well that have kind of the white are like I think they're like oral feeding tentacles sticking out. You can see two of them to the left of center right now, sitting on top of a rock, white. And there's a lot of other stuff living on these rocks too that we can't see well from this distance. But if we were to, when we're done with the transect, and if we find an area where we want to look more closely at some of these things, you would see all sorts of animals that are living in close proximity to the rocks on the surface of the rocks as part of this kind of brown film that we can see right now.
So Ben, how far have we gone on this transect?
Yeah, we have about just 10, 15 meters left to go.
Okay.
Yeah, then we can hold, search around if you want.
Lisa, while we finish up, do you want to explain why we're doing transects? In case anyone's interested to know.
Okay. The reason we're doing these time transects is you can see that the laser is on, and we are going across a standard length of the seafloor, and the lasers will allow us to estimate the swath of area that we are capturing, and then later these videos will be analyzed by experts who can identify all of these species that Jen and I are having a hard time IDing right now, but they know how to do it. And they will then come up with estimates of the number of species that they find in a certain length of the habitat. And then we can come up with abundances or densities, and we can analyze them and see if there are differences according to the type of habitat that we're in, rocks or mud.
Yeah, not to interrupt, but yeah, we're at 200 meters now if you want to call that the end.
Yes,
okay,
let's
great.
do that. Thank you. And by having these standard time transects, we can then compare them across different regions, different habitats, and we can make hypotheses about why are we seeing these differences. Is it because of the current that is in the area, or the depth, or the amount of oxygen, the pressure, all of these physical parameters? We can see how they influence the type of biology that we see. Anything to add, Jen, to that?
So we still have a ship move for 17 meters. So science, really quickly, so you want us to kind of look around, look for some maybe opportunistic samples, and or kind of go into a more exploratory mode right now?
Yes, please. And I think what we're looking for is during this watch, we're looking for a great outcrop of some corals with sponges over which we can then do a 3D pass of the camera, and then that we can then make a model out of it. Not that different from what we did with the whale fall, but this would be much smaller over a boulder that has maybe some amazing stationary species, because obviously if they're moving, that would be difficult to capture them.
So to pirouette.
Does that make sense?
Structure for motion.
Roger that.
Yep.
Yeah. Okay, so we'll just keep on going up to this next waypoint along this ridge until we find that candidate for a photo mosaic?
Yes.
All right, sounds great.
And if you want to mosey on, a little west to the...
Sure.
Just to see the ridge itself.
Yeah, we could do our next ship move that way if you like.
Yeah.
A little bit. What are you thinking for it?
I was going to bring us back to 315 just for maybe 30 meters.
Okay.
Let's see. There was an overhang before. I don't see much of it anymore.
Yeah, the overhang should be a little off to my left there.
Yep.
Oh, see that rockfish right there to the left? Can we just zoom in on it, and then maybe the fish people can tell me what kind of rockfish that is? To the left. Did I say the right? I meant the left.
Yeah, are you talking about the one center screen?
Yeah.
Great. Go ahead, Marley.
It's almost underneath. So if it has... So Di, if you're watching this, what kind of rockfish is that?
Marley, you want to come wide? Kim, there was a heading change to 315. I need to come back over to you now. Thank you.
Pretty big. Thank you, Marley.
No problem. Anytime.
I think a boulder candidate will probably be along this, kind of the larger, because an overhang.
Say it again?
I said I think they're looking for a bouldery type candidate, which might be on this surface
or
here.
Flatter, yeah. Well, I did see on the last watch they have this big outcrop of sponge and coral overgrowth. So.
Roger.
It did seem a little less larger features on the flat part of that. Just thought we'd explore around.
Okay, so that rockfish that Marley zoomed in on was a black gill, and I guess I was supposed to look for a black on its operculum, that is the gill covering, and I guess I didn't see it. I need to have a better eye for that. But we're passing some paragorgids. Those are the classic looking corals that look like branches, 2D branches.
Yeah. Can you hear me now?
Look like cactus, like the ones from that you would see in New Mexico. Yes, I can hear you, Jen.
Oh, good. I don't know why, but I could hear you guys. You weren't hearing me, but I'm back. Yay. I missed out a little on that rockfish tutorial while I was troubleshooting this, but unfortunately, right when we came off transect, I saw a fish off to the right that was swimming with its pectoral fins. And I was like, can we pan right? And no one could hear me. So I didn't get to go look at it. Oh, well.
No, we were
I'll
not
keep my eye
ignoring
out
you.
for
We
it.
couldn't hear you. So, and then these crustaceans that have these long arms, are those squat lobster? Jen,
You know, we saw
you know the one you're
some. I do. So yeah, was that all my days are starting to blur, but there was a bunch of crustaceans that looked a lot like that were on black corals. And I remember that in the chat they were not squat lobster per se, but they were also said to be mostly always seen on corals. So these are on the seafloor. So I'm not certain.
Yeah, those are squat lobsters.
Okay.
Oh, and
Okay.
we're just passing a heterocone or heterocone sponge. They're the ones that have the goiters, very medieval. So squat lobster, I can't help but think of that song from the
Rock
B-52s. Yeah.
Squat lobster. Now I'm not going to hear that song and not think squat lobster instead of rock lobster. So that's fun.
Every single time I see them, that's what I think, and I can hear the doon 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 doo 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 doo. So,
while we're looking around, is this? Can we get any closer to the seafloor just to get a little more clarity on some of these animals, or is that too close?
So
for
this would be probably the closest I'll fly without scraping.
Yeah, no, I don't want you to scrape anything. I was just.
Yeah, no, this is fine.
I do have on my glasses, but still.
Sure.
Yeah, but maybe if we see
this
something
helps.
interesting, we can just stop and then just look.
Sure thing.
And you won't scrape.
Roger that.
Well,
So,
Kurt Whitmer is talking to the Google. He's talking to
go
Google.
ahead, you read it.
And he said that squat lobsters occur in two separate superfamilies. Galathioidea and Chirostyloidae. At least that's what the Google tells me, he says. So
and we have it. We're seeing a lot of them right now.
Mm-hmm. And if you're not sure what we're talking about, one is just passing by on the right. At least I know there's different legs on the video, but you really see these long front appendages with the pinchers compared to, say, a typical crab. They're hiding under rocks, so they are kind of, like rock lobsters.
Yeah.
And we're seeing various white and yellow sponges of different species that have different shapes. And then we're seeing some fragile pink urchins, some different white and yellow sponges, some sea stars. I did see a kind of typical looking sea cucumber. We also have these sea cucumbers that are more sedentary, the white ones, that look almost more like a feather star. We see a number of them right now, but they're not. They're actually sea cucumbers that attach themselves to rocks. If Steve was on shift right now, he could tell us all about them. I guess he spent a lot of time studying and sampling them. He told us on a prior watch about that.
Ooh, look at this really amazing shaped yellow sponge that we're passing on the right. Looks like a decapitated torso.
Wow. Halloween's coming.
Yes, it looked like one of those mannequins without a head. Of a strong guy, a strong mannequin. And I guess this white sponge that we're passing might be called an IKEA lamp sponge.
So I'm checking my videos because you and I are seeing things definitely at different times. And I was trying to decide if I'm behind or ahead.
Huh.
And the white sea cucumbers are called Solus. So I'm just pausing and refreshing my video feeds here.
Yeah, that can be a little annoying when you're not sure what people are referencing when. Oh, Dover Sole. I guess I missed it.
So I'm seeing the Dover right now. I'm curious.
Oh, right now. Yeah.
So, okay, I'm a little ahead of you, I think, now. Have you refreshed your feed lately? Because I just jumped forward a little bit by doing that.
I'll do that.
Ooh, look at this rock. Can you see the cool rock that has a bunch of sponges and a coral on top yet?
Marley, gonna do a quick zoom?
Yep.
That's great there.
So there's at least 3 different species of sponges here. A bright yellow sponge, a lighter yellow sponge, and then the white face sponge. Then
All
one
right,
of
go
those
ahead and come
paranoid corals.
If
And then
Lizzie's on, this might be a good
Yep.
place to do the 3D mosaic. The
It would
photo
be good to know
mosaic.
if this is big enough for what she wants.
It seems, yeah,
'Cause,
relatively small, this rock. We can look for a better
Okay.
candidate, or we
Okay.
might be getting
Okay.
into more of these rocky feature, or these kind of more consolidated larger rocks. But it's your guys' call.
No, I agree. I didn't realize how big we were looking.
Yeah. Yeah. A good reference is the bumper. So if we were to look down at the bumper, oops, sorry. I'm just bouncing you guys all around with the HD cam. Sometimes that's a good reference for us to see how large these rocks can be. So that one seemed a little bit small.
Okay, yeah, Lizzie's agreeing that that's the right concept, but she's hoping for something larger. So we want that mix, I think, of sponges and corals to make it really interesting and diverse, and we're just trying to maybe get a feature that's larger than that.
Roger that.
But it did have nice clear, like we need to be able to get around all sides of it, which can get interesting. So we'll have to keep that in mind.
Interesting feature up here.
This is also an interesting.
Oh, sorry.
Go ahead.
What's the approximate size of the rocky feature that they'd like? Is it around like 3 meters or so is a rumor that I heard?
Vertically.
I haven't heard a rumor that specific, but I do like specific. Let's see, Lizzie is active in the chat. I can see, do you have a minimum size you're looking for, Lizzie? I think she has a few that were the size of the one that we kind of were just discussing. Okay, up to 5 meters. So let's say between 3 and 5 meters would be a good size for a test run.
Roger.
How does that sound?
Sounds great.
Okay, great. So now we're getting into a different type of coral. These
Can we stop here for a second? Or just pause?
Chad, you're not
Chad.
on SVN.
Yeah, I can hear you, Chad.
Do you want us to stop?
Oh, there's a beautiful example of a paragorgid, the bubblegum coral. And then there's a surrounded by white ones.
Chad, were you speaking on
Yeah, I
SVN?
think I had reversed my mic. I think it was, for some reason, it wasn't coming through. Just behind us, I thought I saw some primnoids, but they're a little behind us now. They're little white fan-like corals.
Oh, Roger. Let us know if you see another one coming up and we can zoom on it.
I tried to say something too about those corals and V-Link shut me down.
You no speak.
So, well, neither did you. I guess we were not supposed to look at those particular corals.
Let's hope we find them again. Are they right here in front of us now?
I think this is a little different. This looks to me like a black coral.
Those are, yeah, those are Christmas
like little
tree
black corals. Oh, what's this feature we're approaching here?
About right below us,
Wow.
Jess, we just passed over him.
Yeah.
Hmm.
Little
Can we go back?
fuzzy black corals, I believe.
All right, we're done with the ship move.
Yeah.
Yeah. Yeah. Oops.
Roger. Yeah, I can do a really quick zoom, but I have the ship moving.
Yeah, I know you got the ship moving, right? Okay.
Yeah, the ship move's done.
Go ahead, Marley. All right. Oh, great. Well, then that works out pretty nicely. Sit down, right. Well, it's not going to be a happy sponge there anymore.
Oh, is that a little sculpin? And we've got a squat lobster in the lower left.
Yes, and these are, but those big pectoral fins, wow, it almost looks like it could be a thorny.
Love that trick. Marley, you can go in tighter if you like.
Sculpins.
Yeah, I love the sculpin. It's great.
And then right behind it. And this is a very small example of a Christmas tree coral, the black corals. Corals are long-lived and they're very fragile, but if they are able to grow for a long time, like 300 years, they can become about 3 meters tall, like a real Christmas tree.
Maybe
a big one for your house.
Yeah, I was about to say maybe we can go like 3, 4, 5, something like this, because it seems like if you can see where I'm at there, that's kind of the preference for this ridge.
Yeah, let's do it.
Can you put the lasers on just for a second so we can they can get a measurement later?
Sure. Kim, would you mind turn on the lasers, please?
And can we pan down just a little bit to get a really great shot? This is really cute with the sculpin and the Christmas tree and the. This could be a Christmas
no offense,
card.
Ben, but I'm gonna say that this sculpin is cuter than a hagfish. I'm just going out on a limb.
Yeah. And lasers off, please.
He is pretty cute.
Would you mind turn the lasers off there, Kim? Thank you. We can try putting the porch light on too and seeing if we get more color to it.
Yeah, let's try
that. Sounds great.
Oh. Not too much. Never mind.
Oh, is it? Did you put them on?
Yep.
Oh, I couldn't tell at all.
Roger that.
Still cute. Nice picture.
All right.
Yes.
So.
He is so cute. Yeah.
Guys, I have the telestrator back here, so I put a little hat on him for people here in the control room.
Also, over to the right, there was a really nice, larger.
So,
okay.
Do you want to do?
It looks like.
Oh, I think it's right behind the arm.
And I was just you want to stick lock me here? Thank you.
There's just a larger version of the Paragorgia.
Oh yeah, off in the distance.
That I was gonna, but it's okay.
We're switching out for dinner. See you guys in 30.
Okay.
Okay. Let us know what you're having.
We just have meal relief happening right now. We'll resume momentarily.
Yeah. No, it's an important component of vessel life. You do not, meals do not wait for you, and you need to be timely in getting them. It's, I was saying to someone that leading some of these cruises from home is very different in many ways, but one is that no one is making me any meals. I have to do that for myself, and that's something that I miss, for sure.
Yeah. The most important person on this ship is the cook. Can really set the tone and morale.
What are we doing here?
Yeah, so I just want to bring us a little north to keep on this contour. So I'm going to bring us maybe just 40 meters, 340, and then we'll head a little more to the west.
Roger.
Here's running. I can put that in now.
Sure.
And Lisa, I recall hearing a request for 1 to 2 more transects total.
Oh.
Was that what you heard? Ooh,
Yeah,
look
1
at
or
that
2.
little red guy.
Oh,
Okay,
can
so we've
we,
already done 1.
Do we have to keep? We have to
Can
move
we-
right now.
We don't have to.
No?
There's this
can-
guy down here. I
what are you
just want to take a
thinking?
look at if we can.
The red fish?
What do you want to look at?
What is
It's just off
Yeah.
to the lower left now.
lower left?
Right here. I got the telestrator. There we go. I know it keeps moving, right?
The sponge you want to look at?
Yeah, it's just a sponge. We can move on.
Or there?
It's okay.
Let's move.
Zoom in? You want to zoom in on that white one?
No, it's okay. We can move along.
Roger.
Is that a basket star, that the arms that we're seeing in the lower left? Looks like it.
Lower right?
Lower
Just
right.
go-
Oh, God, yeah. Exactly. I'm
Got
the
it.
worst. The worst.
The flattened
There's
one
going to be
there?
a lot of that. Is it
Okay.
your left or your right? Yes.
It's coming
guys
into
have
the
to-
center, so that's
Put the mics a little closer to your mouth, I think, because some people are really loud and some people are really quiet.
So which one is too soft?
You're quiet.
Me? I'm too
that's
quiet.
Lisa.
Yeah, you're quiet. I
Wow.
can barely hear you. And then somebody else is really loud.
That's probably me,
Oh,
Jennifer. Or no?
So Kim, tell me, this is Lisa again. Am I too soft?
sorry. Let me
Yeah,
check.
a little soft, yeah.
Oh, wow. Should I take the sock off? Would that help?
Yeah, that did. So
Okay.
what are we doing?
Does that sound better?
It does, thank you.
Okay. Thanks for telling me that, Kim. It's very important that you can hear me well.
Yeah. Okay. Which one are we zooming in on? Is that what the intent was, or are we?
I think we were done.
Oh, okay. The ship move is in 340, 40 meters.
So moving on.
Yes. We are on the hunt for a good target for a 3D video kind of like we did for the whale fall. We're looking for something 3 to 5 meters.
We just did one on the last shift.
Well, they want another one that's
Another one.
3 to 5 meters size. We're looking for it to have a mix of corals and sponges. Of course, we need to be able to get around all sides
Roger.
at different altitudes. So that's what we're hunting for.
3 to 5 meters. I see a target 30 meters away.
Okay. Lead the way.
We're coming along for the ride.
Yeah, me too.
Yeah, I love that you can see that far ahead, Kim. Awesome.
I don't think that's Kim, though. We had a change out for dinner.
Oh, this is Dan sitting in for Jess.
Oh,
She's down in the chow
Dan
line.
then.
Oh.
Sorry, Dan.
Well, that's okay. I answer to anything.
Sorry.
Don't say that.
I've
You'll regret.
been married for 25 years, so.
There's our feature. Oh, yeah.
That is the
So
feature.
you guys,
Might be bigger than 3 to 5 meters.
oh,
Yeah.
look at that. Okay, Lisa, can you see channel 3?
Yes.
So now we've got, can you tell us which is on the right and the left on camera 3?
So we've got the sonar
Or
up there
channel
right
3?
now.
Mm-hmm.
Yeah.
So I think it's the Hercules sonar is on the left,
That's
Yes.
correct.
and the Argus sonar is on the right.
You can't see the target anymore because
Yeah, I just had.
we're there.
But it's interesting to see. Thanks for putting, that's Steven, right?
Yep.
Thanks for putting that on. It's really interesting for us to get a chance to see that.
Sure.
I've never seen that, so it's very cool. Thank you for sharing.
Yeah, I was trying to show you when they
So-
when they spotted the target 30 meters away. I was trying to show you what they were seeing.
This to me looks like an alligator head.
Pretty cool.
Oh, I see it. It's a pretty good description, yeah.
Yeah.
So are
What do
we
you see in the
Oh, I
clouds?
see it now.
Yeah. Watch out,
commencing
Captain Kirk.
360 or what do you guys want to do?
Okay. Lizzie is liking the height of it. Let's take a one circle around and make sure that there's nothing that will get in our way.
I'll come closer to you.
Roger.
Does that sound good, Lisa?
I can.
Yeah, it sounds great.
I can tell you now that we're good to go around it. There's nothing in the way.
This is, yeah, and this is a lot bigger than what I thought you wanted, Lizzie. So I'm glad you're here to say yay or nay.
The other side is appears to be a cliff, so.
So Lizzie
It's
said
interesting.
yes, let's do
Liz,
it.
okay, but she was also saying, is this something, and I like to think of it from that perspective, is this something that if you saw it as a member of the public, you'd say, I want to interact with that. I want to look more closely at it. So far, I think it's pretty interesting looking, but it's good to think about it from that perspective of,
I don't know.
like when you see the whale fall 3D, you definitely want a chance to move it around and zoom in and zoom out. I think that's what we're looking for.
Just needs a nickname.
Yeah, this is not exactly whale fall quality.
Okay.
Well,
all right. That's a high bar,
Yeah,
but
but
still, it's good to think about. What if we call it Alligator Rock?
Yeah, see, there you go.
Does
That's
that
what
make
it
it-
needs.
Alligator Rock, that's a good one.
yeah.
Then it's like, oh, okay. I'm interested in that.
There's no giant corals on it though, so.
True. It's
Yeah, so the besides that, also resolution scales with how large or small the object is. And this being
Okay.
quite large, there's going to be a limit to the number of points you have on the wireframe and the resolution of the photos draped over it. And because the corals are relatively small to the head itself, the alligator's head, if you will, those corals aren't going to show up very well.
okay.
They will show
right.
up, but they're not with great detail. So yeah, for 3D mosaic, I don't know if it's the best target just because of that reason. If you had some of those Davidson size bubblegum corals and like,
Bubblegums.
yeah, large Christmas trees or something like that, I think it would be a much better target.
Okay.
Okay, that's
moving
good.
on.
We're learning. We're learning target selection. I like it. That coral over there is like, what about me? But sorry, not enough. Not enough buddies with you there.
And we're headed towards 83 in general, I'm thinking.
Yeah.
Mhm.
83 and then towards 84.
Roger.
But we have a lot of leeway on that. As think of those as
Yeah.
suggested directions. If we see an interesting target, we definitely can take a look at that first.
Roger.
Yeah,
Sorry
we're
to
just
make you
following
dizzy.
it.
Centering the camera.
No. But we're in a general direction of 84. But as Jen said, if we see something interesting, totally okay to go off.
Okay.
Copy that.
I'm going to generally follow this ridge line. And then let me know if we see anything in sonar. Which will be roughly... I guess we should get a little closer to it.
So we have now Dan as the Herc pilot, or who's speaking now?
That's. Go ahead, Rennie.
That's Rennie, navigator, and then Dan's Herc pilot, and Jake is the Argus pilot.
Okay.
All right, we're going to be bearing
Rennie.
305.
Roger.
Yes, sorry, go ahead.
Are you the person I've been emailing with about the mapping data?
Is this Lisa?
Yes.
Then yes. Lindsay and I. Lindsay and I, yes. So I've
You
been.
have an You have an interesting first name. My uncle has that name.
Oh,
He's
really?
Italian.
Yeah,
Yeah,
that's mine.
Renato Bertolini.
Bertolini, yeah, that's pretty good.
Yeah.
Yeah, mine is also Italian in origin.
It's a very romantic name.
Renato?
Renato. Yeah, Renato.
100 meters at 305. Thank you.
Oh, okay, we've got the sonars back on. Channel 3.
Yeah, looking for targets.
Nice. So if
At
you're
the end of your
following
leash.
along and say you're looking at channel 1, the Herc camera, and you want to know what we're talking about, you could put on one of the other channels, including channel 4, which will show you the quad view, which will be Herc cam, Argus cam. Right now, 3 changes, and right now it's showing the sonars from both of the ROVs. And then the 4th panel is always a map.
And so what you can see on the 3rd channel, where you can see the sort of sonar imagery, one of them is from the first one on the left. I know this left is from the Hercules, which is the one that's on the sea floor. And the second one to the right is of the Argus. And it really gives you a 360 view of what they can pick up using sonar on different features. So we're looking for a feature to construct a photo mosaic of.
Just for reference, those are always referenced to up is vehicle forward. So they're not north referenced. Their vehicle forward is the up direction, backwards is down. So as they change.
Okay, so right now the targets are forward and starboard on Herc.
That's correct. Yep.
Okay, thank you for orienting for that.
Yeah.
That's helpful. And it looks like there's no, so it must fall off to the port side then. Goes deeper.
Yeah,
target returns over there. Okay.
That sonar on Hercules is sitting on the top, so it doesn't see downslope like that. Whereas Argus's scanning sonar is mounted underneath, so it can see a bit down.
Oh, okay. Thanks for the orientation.
Sure.
$10-meter range rings on the left one and 20-meter on the right. Little red rings.
Well, those are hard to see, but okay. Got it.
Yeah, I need to turn out.
I think it's, easier. The van's very dark inside, so I've tried to darken my room, but not completely.
And if you go to satellite feed 3, you can see that sonar full screen.
Approaching target.
Yeah. Big corals, come on.
It's a rock.
Okay, so we're looking at the scale of the organisms to the size of the rock and trying to have more organisms than rock, I think. This is a little better than the last one.
Yes, I agree.
There's a target 30 meters off to the...
But I would hold out for one with bigger corals. What do you think, Lisa?
Or sponges, yeah. Yeah, I agree.
Okay.
We've also had some luck in the past with individual sponges, individual corals.
Yeah.
Yeah, that overhang would be problematic too, because you probably wouldn't get much of the underside, and so you'd have a big hole in the model.
Okay.
Oh, so Daniel Geiger asked if we can look under the ledge here. So can we stop?
Yeah, I'll wait for the boat anyway. So
okay, great.
Get under
Thank
there
you.
and put some light on it for you.
We're looking for brachiopods. in the sediments under this rock. And these would be, if we find them, for the Santa Barbara Museum of Natural History. Did I get that right, Lisa?
Yes. Daniel is from the museum, and Channel Islands National Marine Sanctuary is partnering with them on these dives.
That's great. Oh, on rock, he said. So
On the rock.
Scan up. So originally I'd heard about we were looking in the sediments, but I guess here we're looking for live brachiopods on the rock.
Let me pan up,
It's
Steve,
very
where
dark
there's
in there.
more light on it.
It's
I
nice
was mentioning earlier, though,
things up close.
if we look closely, yeah, then you see a whole nother kind of layer of biodiversity when you start looking closely.
Yeah, completely different community of smaller organisms.
Don't see any yet. Look at the clams.
Well, they look like
Did you
clams.
see clams? Mm-hmm.
No.
Oh, they look like clams, right. So if we could,
Yeah.
that little squat lobster is so cute. But if we could zoom out some and maybe pan across.
Yeah, can do.
You want me to hold here?
Oh, look at that little rockfish.
Yeah, I think so, yeah. Zoom in there on the cup coral.
Star. Oh,
And
and there's a rockfish. Just based on the color, I would call it a rosy, but probably not. It's very light colored.
Okay. Find the clavularia. What is clavularia? I'm looking at.
Zoom right in one, Steve.
Full zoom.
Roger.
Oh, the cup corals are clavularia.
Mm-hmm.
Oh, a rose thorn at this depth. Okay, I was close. That rockfish was a rose thorn.
Okay, do you see the, I think it's
pretty.
this coral that's to the left of the bat star. There's a cup coral. Oh, that's Desmophyllum, the cup coral. And clavularia was I think I saw that polyp earlier. If we zoom out.
It's the smaller one.
Can we back out a little more and look up and to the right?
Up and to the right.
Yeah, but I'm not seeing it easily now. It was closer to the rockfish. It was little. I think it. I saw something retract that looked like a larger kind of polyp. I think that's what it was. But
what his nose is looking right at it.
Okay.
But no, I don't think we can.
Very tiny.
Ah.
Yeah, do you see it hanging there?
Yeah.
That was the thing that retracted. It looks like oh, so Daniel Geiger saying that the clavularia was to the left.
White to gray, encrusting on rocks with shaggy-looking polyps. That's the description of clavularia.
Moldy.
What's the size of it?
Oh, is it up there on the top? Okay, I see it. So move over.
Yeah, they look like the little hydroids. They're still to the left.
To the left, Mark?
They're, yeah,
tell us
octocoral, I believe. It's this stuff right here. Oops, that's a number.
Roger. I see what you're on about.
I want a stylus.
Okay,
Stephen,
Steve.
can you put the telestrator in 3?
I can.
Thank you. So you're going to get to see the telestrator.
You want to see it in action?
Yeah, draw some stuff.
Yes, please.
All right, here we go. So they have different tools. Here's the draw tool. You can just do freehand, right? You can, this is great for when you want to draw a path for the pilots.
That's how I usually fly.
Kind of nice. You can just circle things. And I was just playing with this one. You can count